Okay, I guess we'll get underway. Welcome everyone to our virtual Pioneers in Biomedical Research Seminar series today. It's my privilege to introduce our introducer, Dr. Flager, who will be introducing Dr. Qian. And I'd also like to extend a big warm welcome to Dr. Qian, and I'm very much looking forward to her talk. So at this point, Jessica, pick it up.
Okay, so it is my great pleasure to introduce Dr. Qian from the University of North Carolina at Chapel Hill. Here, she's an associate professor in the Department of Pathology and Laboratory Medicine and is the associate director of the McAllister Heart Institute. Dr. Qian is really a pioneer in the area of cardiac reprogramming. And what I love about her work is that she really uses a combination of cutting edge techniques and technologies to really understand this process in regeneration and repair of the injured heart. This work has been published in numerous high-impact journals, including Nature and Circulation. She's also received numerous awards for this work, including most recently the Emerging Investigator Award from the NIH and the Established Investigator Award from the American Heart Association. She also serves on the study section and has mentored numerous successful trainees. So with that, we welcome you virtually to the FBRI, Dr. Qian, and we're really pleased to have you with us and we look forward to your seminar.
Yeah. Thank you, Jessica. Thank you, Robert. Thank you so much for the invitation, and I will share my screen, open my slide. All right, so I guess I will just get started. And yeah, it's such a great pleasure to be here. It's, you know, as Jessica mentioned, and I will really share with you the work we have been doing and we are doing now, and also give you sort of an overall picture, maybe a flavor about what, you know, how we get started, how I get started as a scientist to really get into the research of this very fascinating organ, the heart. So I think I'll get started from the very, very beginning. My passion in the heart was really kindled by this very kind gentleman, Rolf Bodemer, showing this picture. And at that time, back to the late 1980s, he started his lab at the University of Michigan and was really looking through thousands of the fruit fly Drosophila melanogaster embryos in an effort trying to identify potential regulator in the nervous system. And accidentally, he happened to see a gene that is active in the heart. And instead of really tossing this away, he pursued with full curiosity, even generated the mutant embryos lack of this gene. And astonishingly, he finds the mutants, they do not have a heart. He called the gene T-man, really following the Wizard of Oz character that lacks of this gene, or lacks of a heart. And soon after, Rolf discovered this work and published in 1993 in Development, and there are many other scientists and researchers followed on this interesting phenomenon and identified the human homologue of this gene. And now you guys probably see, get very familiar with this gene. This is the NKX2.5, and has a wave of study led by Seideman's group, Ken Chian's group, and Richard Harvey's group. And importantly, they discovered that the mutation of this NKX2.5 caused not only congenital heart defect, but as well as the conduction defects in adult heart patients. And there are a lot of media reports, and the story is very fascinating as a series of almost like scientific fiction. I traced down Rolf and joined his laboratory, very fortunately, and there also with another grad student in the lab, and probably many of you also know him, become my significant other and also the long-term collaborator for the last 2 decades. And there we started to work together, looking for team as friends and how they work together to really orchestrate the development of this very fascinating organ. And during my PhD, I really took advantage of the powerful genetic tools in the fly and learned how the master transcription factors and together with the signaling pathways elegantly choreography the lineage commitment of cardiac cell in a fly heart. And of course, you probably see the trace of my training and sort of the habit, the way of how I do research, because even now, from the publication from my own laboratory, you can see that we really love to study transcription factor and signaling molecules. We also love to take unbiased forward genetic screens, and we love to think about the way how gene works in an upstream, downstream fashion and do epistasis analysis. And we also love to use in vivo animal models. And through the work when I did in Rolf's lab, and I really received a very solid training in developmental biology, and also which really laid a solid foundation for my postdoc work in the laboratory of Deepak Srivastava, who is also a fascinating mentor and support us to really freely explore the ways to regenerate injured heart. There, I actually explored different directions, and including using microRNAs and using stem cells, and also most excitingly, during my late stage of postdoc, and I really go into the reprogramming work and try to identify the condition and cocktail to fix injured heart using reprogramming approach. And of course, my current lab also still continue working on those other directions. But today, for the sake of time, and also majority of our lab's focus is on the cardiac reprogramming, and my remaining talks and slides will be just to focus on the direct cardiac reprogramming. And from the molecular mechanism all the way to potential translational work in the future, but more importantly, how we use the reprogramming as a platform, and also to study the fundamental biology underlying cell identity, cell plasticity, and cell fate determination. That has to also trace back to that original paper where I end my training with this quite nice paper published in 2012, where for the first time, we demonstrated the possibility of using direct reprogramming approach to mend a broken heart. And in a mouse model of myocardial infarction, and we show that the introduction of 3 cardiac transcription factors converted the endogenous cardiac fibroblasts into iCMs, results in functional improvement and the scar size reduction. And using genetic lineage tracing and post-labeling, and we demonstrated that the new muscle tissue around the scar indeed had a fibroblast origin. However, back to 2012, in spite of the excitement, there were also a lot of doubts and questions. And I personally believe that's naturally due to the infancy stage of iCM reprogramming. And I always summarize into 2 categories about those comments and concerns. And the first category is really related to the technical hurdles and concerns. That includes inconsistent reproducibility and relative low efficiency at that time, and also batch-to-batch variation. Also naturally due to the crude targeting of fibroblasts using viruses, and of course, the potential side effect and long-term safety concern. Also back to the 2012, almost 10 years ago, there were also a lot of biological questions were not addressed at all, such as whether those cells are functionally matured and how similar they are to the endogenous cardiomyocyte, and whether there's still a remaining feature of fibroblasts in those converted cells, and whether the conversion involves stem cell program. And when you really look at the fundamental biomolecular mechanism, what are the epigenetic mechanism? What are the metabolic mechanism? And also maybe when you step back a little bit even more, look at the cardiac reprogramming together with other somatic reprogramming, for example, neuron reprogramming, hepatocyte reprogramming. Is there some similar mechanism shared among all the different direct reprogramming? And with those excitement and also questions in mind, I started my own laboratory at UNC Chapel Hill in the fall of 2012, and almost 10 years ago. During the first several years, we made really hard effort trying to better understand the process of iCM conversion. And for the first research area, we leverage the knowledge in developmental biology, where a phase-4 cell fate conversion relies on the delicate balance of transcription factor expression, we characterized the stoichiometrical requirement of the reprogramming factors and perfected the recipe. And in this area, we find out that you cannot just throw in the transcription factor randomly, and you have to find the best optimal dosage and also the timing, and which can also be achieved by using the polycistronic here, and especially encoded by the MGT following the order of MAP2C and GATA4 and TBX5. And in such way, you get a high expression of the MAP2C and relatively low expression of GATA4 and TBX5, resulted in a much improved efficiency for both in vitro and in vivo iCM reprogramming. In addition, we also fine-tune the process by profiling the early epigenetic changes, and in particular, through a unbiased functional screen, we identify the main epigenetic barrier to this process, that the gene called BMI1, and we published the work in 2016 and 2018. And by removing those epigenetic barriers, we also improve the efficiency and yield of iCM and streamline the process of iCM reprogramming. More recently, we also studied the role of autophagy in protein remodeling during iCM conversion. And we show the activation of autophagy during early stage of cardiac reprogramming. And we also show the absolute requirement of ATG5 dependent autophagy. And because once you block this pathway, reprogramming process is completely compromised. Surprisingly, when we targeted additional nodal point and going more upstream of the autophagy, we found that knocking down of backlink-1, a well-described key regulator for autophagy initiation, resulted in an unexpected increase in reprogramming efficiency. And mechanistic characterization led to the discovery of a previously unknown function of backlink-1 in regulating Wnt beta-catenin signaling pathway involving ULK1 and PI3 kinase pathway. And showing here just one representative data, and you can see that with the backlink-1 knocked down during the reprogramming process, and beta-catenin nuclear translocation from cytosol to the nuclei is much enhanced, and leading to the much increased transcriptional activation of the downstream targets. Importantly, in a mouse genetic model of backlink-1 haploinsufficiency, we show that enhanced in vivo reprogramming due to loss of backlink-1 resulted in further improvement in heart function and further reduced scar size. And we published the work now about more than a year ago. And during these years, we also generated many tools for the community to extensively study this very promising and interesting approach for tissue regeneration. And we deposited the most popular and frequently requested plasmid to the AdGene. Actually, last year, we deposited another dozen. So just keep on checking our website. And if you have any question, in terms of their map or the usage of them, just, send us an email. We also continue upgrading and updating our protocols for both in vitro and in vivo reprogramming. And nowadays, we always use like Zoom and Skype, and can sometimes provide also live training for the people who are interested in these protocols. We also love to solve puzzles, and especially the students in the lab. And our recent paper on this isoform-specific effect of MAP2C, a core reprogramming factor that almost appear in all current reprogramming cocktail, really provided some answers to the seemingly inconsistent result from different labs using even the same cocktail. And in that paper, we basically collected the reprogramming cocktail from different labs and find, oh, wow, even MAP2C, different lab using different isoform. And that isoform really give the different reprogramming results. And, feel free to go into the paper and find out how the puzzles get solved. And also we deposited the different MAP2C isoform in combination with GATA4 and TBX5, and sometimes with HAN2, and using these tools to really tease apart how the different isoform of MAP2C give rise to the different reprogramming outcome. Our refinement and understanding of the cardiac reprogramming has been benefited also from the single cell omics approach that really evolved very fascinatingly and in a very quick way, which really addressed the challenges due to the two inherent features of any reprogramming process. So that is asynchrony and heterogeneity. And a few years ago, by using the single-cell RNA-seq and mathematical modeling, so our very talented postdocs Qian and Li Wang, and also grad student Josh Welch, reconstructed the molecular route of how a marine fibroblast become a marine iCM. And they demonstrate the stepwise transcriptional shift along the process and identified and characterized the intermediate cell population, including Fib, iFib, pre-iCMs, and finally the iCM. And importantly, they didn't stop there, and they went back to this route and really took advantage of the unprecedented high resolution to answer many long-lasting but difficult to be answered questions using traditional approaches in the field. And hopefully they provided some answers to the questions I listed here. But I want to really highlight one finding, which was kind of surprised, but also extremely rewarding, starting with how we take advantage of this new approach and new tool. So where, as I mentioned, we use the single cell RNA-seq and mathematical modeling to reconstruct the route of the conversion. We simulate the process and also experimentally validate them. Then by pathway analysis, we identify the new gene clusters that change along the process significantly, and especially the cluster 3 caught our attention. So that's the down-regulated and transiently upregulated genes. And Ziqing, Li Wang, and Josh pull out those genes by the geo-analysis, and they didn't stop there again. So they find out the 24 candidate genes that belong to all the categories, especially for the geo term, highly enriched the RNA processing, splicing, and splicing factors. And this 24 this 24 splicing factors, and it sounds very interesting in their identification, and they start experiment. And they did both the gene expression analysis, trying to find how these 24 candidate genes are differentially expressed in cardiac fibroblasts versus the cardiomyocytes. And at the same time, they also took a functional screen approach and screen out these 24 candidate genes. And from both approach, the top candidate actually is the same. So this is the splicing factor PDBP1. And by further analysis, they validate that removing of this splicing factor actually enhanced iCM generation. And by looking at the underlying molecular mechanism, PDBP1 and really serve a role to regulate the splicing repatterning. So remember here, it's not transcriptional repatterning, it's the splicing repatterning from the fibroblast to cardiomyocyte. And they also identify the downstream targets and the geo terms regulated by this very amazing splicing factors. So for the details, I think we spent one figure on this very surprising but rewarding finding regarding the PDBP1. And currently, my grad student actually generates the PDBP1 cardiac fibroblast specific knockout and want to tease apart its in vivo role in maintaining cardiac fibroblast homeostasis in the adult heart, and how that responds to the disease condition, and how we can learn and leverage the knowledge from there to further improve in vivo reprogramming using the tools and the mouse models we generated. And just, hang there, and we will probably report to you guys more in the near future. And 2 years later, another very talented postdoc, Yang, who actually is assistant professor at UAB now, built the molecular route of the human cardiac reprogramming at the single cell level. And slightly different from the mouse work here, she actually took a approach that is to profile that in a time course way. And she collected the cells at different time points during the human cardiac reprogramming. Also, she used a more advanced analytical pipeline, and this time combined the slicer algorithm together with the RNA velocity, and she found out a decision point where a human fibroblast either decided to go to become a cardiomyocyte or go back to its original fate, that is a fibroblast fate. And he also introduced a new algorithm in this study, which is called cell fate index, which really allowed the compression of the whole genome information into an index number for the comparison of cell fate transition between different species or under different conditions. And we believe this cell fate index actually can be utilized to evaluate the progression of different cell fate conversion, not just during the reprogramming process, can also during like development process or other cell fate conversion processes as well. Through this work, we believe we sort of created a crude or rough Google map for reprogramming, where a fibroblast is analogous to a car set to go home, that is a cardiomyocyte fate. And our trajectory map sort of guide the car to avoid wrong route and the refractory route I just mentioned on my previous slide, and also the roadblocks that could be some of the epigenetic barrier I mentioned earlier, and also with the helps from hospitals and gas stations. That's potentially the genetic or epigenetic facilitators during this process. And our early work actually built this rough Google map primarily based on the trajectory using the single-cell RNA-seq approach. And more recently, we start to refine the trajectory by incorporating the epigenome information at the single-cell level. So this study just was published a few months ago, where the postdoc Hao Fei Wang and another talented postdoc Yu Chen, who also became a faculty member in China, and they actually performed the single-cell ataxic to characterize the chromatin accessibility dynamics of early iCM reprogramming. And they performed an integrative analysis together with the single-cell RNA-seq data, and which really allowed them to identify new transcription factor, and in this case, SMAD3, potentially regulate the early fate conversion. And there, they found this SMAD3 actually play a bimodal role. At the early time, it sort of inhibits the conversion, but later time facilitates the process. And in addition, they also discover a global rewinding of the cis regulatory interaction of cardiac genes along the reprogramming trajectory. And in the paper, they showed two examples where for the first one is sort of the gained enhancer promoter interaction, and ACTC is the examples they showed in the paper. And for the other example is they call the switch mode, and where the promoter, the enhancer promoter interaction switch from either the inhibitory or the activation to another one. And they showed in the paper is the Titan gene. And actually not just by the single cell double dual omics data they showed, but they also validate with all the chip experiment. And this work really demonstrates the feasibility of using the single cell ataxic, and especially the integrative analysis using the single cell RNA-seq we published early to identify new regulatory transcription factor involved in the early stage of iCM conversion that could be also targeted for the manipulation to further improve the efficiency and quality of iCM generation in the future. And as you can see, single cell genomics approach always yields tons of data and pretty challenged data to be analyzed. And at the same time, our lab also has a group of people dedicated to the dry lab part. And we continue also contribute to that as well. And we develop some of the algorithm in collaboration with Josh Welch and Young Prince. And the slicer is one example. And we continue actually upgrade the slicer into spacer. And a few years ago, Josh actually published the library, which is an even better version compared to the early one. So that's almost 2 or 3 years in cell. And more recently, we published this work. The new algorithm is called topologizer, which is really allow the characterization of the topological relationship of the subpopulation during a biological process. And the work was last year on publishing EMBO reports, but I wouldn't spend too much time. And because that's actually utilized in heart regeneration in zebrafish, and perhaps you guys will hear the talk from either Hong or like Jiandong at other meetings or other occasions. But we hope to continue building or upgrading the algorithms, incorporating more and more single cell omics approach. And you guys probably realize every week, probably there are new paper coming out. So that includes the single cell ChIP-seq, and people also call that co-batch. And more recently, the single cell Hi-C was also available. And it's very interesting, last year at ISHR meeting, like all of a sudden the single cell RiboSeq was out, and I was like really chatting with the people at the breakfast table, talking about how this technology can really help us evolve, address the questions remained in the cardiac field, but at the single cell level to look at the translational regulation as well. It's really exciting. But coming back to the map, I talk. So we talk a lot about the map and the different routes and different roads, how we can avoid the roadblock and going on the right path. But we all know that a good performing map also needs a superior car in order to get back home fastest. So I sort of put the car analogies to fibroblasts, but looking at fibroblasts, we know the complexity of fibroblasts and especially the heterogeneity of those fibroblasts in vivo becomes more complicated with disease progression and aging. So how to identify the best cars? So we decided to really spend more efforts and try to find out the features of these cars, and perhaps identify our gene Tesla in order to refine the in vivo iCM reprogramming, especially under different physiological and pathological conditions. And a couple of years ago, we again, took an in vivo approach and modify how we isolate the cell and really take the perfusion method. We hope to keep the cells at relatively healthy and physiological condition, and really not to disrupt their nature features too much. And our improvements also include the high throughput way, and we sequence a lot of them. And we also optimize our data normalization and batch effect correction at that time. And of course, so no conclusion will be valid only if we experimentally validate it. So that's always the thing I said, how you are going to use the single cell omics approach. Don't forget to validate some of the conclusion experimentally. So again, this time we also use the single cell dual omics approach and to looking at all the cardiac cell, except the cardiomyocyte, all the cardiac non-myocyte, and using this approach. And we perform the unsupervised data analysis, and we characterize subtypes for each non-cardiomyocyte major cell type. Including fibroblasts, including endothelial cells, and also immune cell and parasites. But we primarily focus on the cardiac fibroblasts, and we sort of regroup the fibroblasts, or we categorize them into 3 new types of fibroblasts. And we also relate those 3 groups of fibroblasts to their functional state. And I'm, sort of try to summarize the study here because it's also just published. And we believe our group 1 fibroblasts more responsible for the cellular response, especially to the external stimuli or injury. And the group 2 fibroblasts more responsible for the cytoskeleton organization, while the group 3 mainly responsible for immune regulation. And we think the group 3 is probably the fibrocytes people already studied for years. So that's why we are primarily focused on the group 1 and 2. We also perform some very preliminary functional characterization and using the adult primarily isolated cardiac fibroblasts and knocking down the group 1 and group 2 top candidate gene and to study them. And here we actually, one gene is called HSD11-1B1 and the other gene is the GFPT2. And they're highly enriched in these 2 different subgroups and also exclusively expressed there. And when we knock down these two genes, and we find the functional phenotype actually explain the geo terms we identify using the single cell omics approach. So we are very excited about these two candidate genes, define these two subgroups. And we also look at how these two groups of fibroblasts respond to MI. And we find group 1 fibroblasts, they respond in less dramatic way than the group 2. So we sort of start to bet a lot of money on the group 2. And we know that's not the end of the story, although the leading author also left the lab, started their own laboratory, and they're very exciting new career. But we think it's just the beginning. And we are going to further characterize each state. And in particular, we already generated the flux allele and also pre-ER allele for the top candidate genes, especially in the group 2 fibroblasts I just mentioned, the GFPD2. And not only we are going to study the function and do the lineage tracing, but more importantly, we will also very specifically to lineage deplete those cells and to see their requirement. And after that, we are going to target those subpopulations of fibroblasts for direct reprogramming and to see whether that's the right choice. All right, so far, this is all published work because I also want to save a lot of time for unpublished work. And here I'm going to sort of a little bit switch the gear, but not completely, and tell you some very unexpected finding, but also very exciting in another way. So like coming back to the paper we published and using the single cell dual omics to identify the transcription factor highly enriched in different cell type and different subgroups of one particular cell. And we focus on the fibroblast. And there is a very interesting observation caught us attention. So when we determine the promoter accessibility of the transcription factors across the major fibroblast subtypes, we identified putative binding motif for 42 transcription factor enriched specifically in fibroblasts. And interestingly, we find how many? About 50% of those corresponding transcription factor belong to the BHLH family. So what does that mean? Basically means in the fibroblasts, there are a lot of open, accessible chromatin area, allow the binding for BHLH family. And just keep this in mind. And also we find 7 of them exhibit high expression in fibroblasts and including again 6 BHLH family members. Just keep this in mind. And now I talk about another project. Haofei, postdoc in the lab, and Ben Keepers, a very talented MD PhD student in the lab. This is in a separate project where Haofei and Ben explored possible shared mechanism among direct reprogramming. And they chose the 3 reprogramming system representing 3 different germ layers. The IHAP hepatocyte representing endoderm, and iCM representing mesoderm, and ineuron representing ectoderm. So at day 3 of reprogramming, so Haofei and Ben explored the enhancer dynamics as well as the super enhancer changes, the enhancer promoter interaction, and also in combination with the transcriptome analysis. And of course, at a much later time point, day 12, and they validate the reprogramming were successful using the cocktails here, and also the cocktail already published years ago. So when they took a look at the gained enhancer, well, it's not surprising the neuron will gain neuron-specific enhancers, cardiac reprogramming will get cardiac, and hepatocyte will get hepatocyte-specific enhancers. But one thing to note is that during the IN, the neuron reprogramming, so what did you guys see? So they find that apart from the enhancers related to neuron function, the enhancers related to muscle program is also being gained in IN reprogramming. Not only enhancer analysis, so Haofei and Ben also look at the super enhancer. Again, for neuron reprogramming, they saw enrichment of muscle-related organs consistent with the observation with their enhancer data. So what's really going on? So they went back to look at the cocktail and did all different experiments. And they find out that this aberrant activation of cardiac program was in fact caused by this neuron transcription factor called ASCL1. So it's one of the cocktail. And interestingly, when ASCL1 is overexpressed alone in fibroblasts, apart from the neuron-related biological process, they observed the enrichment of muscle-related geoterm. And furthermore, so they also did the qPCR to confirm that. And the most astonishing is here, the RNA-seq data. So when they compare the ASCL1 overexpressed fibroblasts to the fibroblasts infected by reprogramming factor, the classical GMT or the classical GMT plus HEN2, and they find the ASCL1 can also upregulate many genes that are being upregulated by GMT or GMHT. But at the same time, ASCL1 can also upregulate another panel of genes, actually quite many genes, that fail to be activated by the traditional reprogramming factor, but are highly expressed in the endogenous cardiomyocyte. So with this kind of data in mind, Harvey and Ben decided to do something really crazy, to use these neuron factors to see whether it can maybe help or maybe can drive cardiac reprogramming. I will probably skip a lot of the data, but just really use this key data to show, finally, they find out ASCL1 indeed can be used as a cardiac reprogramming factor. And when they add this ASCL1 into the classical MGT reprogramming cocktail, they saw a significant increase. And moreover, when they withdraw a factor at the time, they find out ASCL1 just in combination with MAP2C actually is sufficient to convert fibroblasts into iCM. And not only true in the mouse system, but also true for the human cells. And this A and M reprogrammed iCMs exhibit calcium constellation, action potential, ion channel activity, and also gene program shift from fibroblast to cardiomyocyte. At this time point, you might wonder how this could happen. Is that simply an artifact? And why a neuron factor never expressed or never being studied in cardiac system has such a powerful cardiogenic potential? So in fact, we talked to different people around the time. We are also very puzzled, and I even call back to my PhD advisor. And ASCL1, has that ever expressed even in a lower model organism, fly, zebrafish, or any lower model organism ever in cardiac lineage? The answer always no. So it's a clear neuron factor. So then maybe we have to go back to the textbook and look at what is BHLH factors. And this is a transcription factor family, referred to the ones that have the basic helix loop helix structure in their DNA binding domain. And they usually form homodimer or heterodimer to bind on the E-box motif of their target sequence. And many well-known transcription factors important for development are actually from those BHLH family, including ASCL1 for neuron development. And another one, then the cardiac people will immediately know about it. That's HEN2 for cardiac development. I also mentioned that HEN2 has been used as one of the reprogramming factor in some other cocktail. Importantly, by structure analysis, Harvey and Ben found that the basic helix one arm of the ASCL1 is intrinsically much shorter than the HEN2, really allowing the more efficient binding of this BHLH transcription factor on the nucleosome. And then furthermore, they also performed the single cell analysis and show that when comparing to the traditional MGT mediated reprogramming, this A and M mediated one exhibited a new iCM subpopulation that strongly activated a group of cardiomyocyte structural and functional genes. And they further reconstructed the trajectory and now find out this A and M led to another two new categories, two new trajectory. One is the trajectory 2 and 4. 2 is more to the neuron fate, but that can be corrected later on. And this trajectory for really going to a more matured cardiomyocyte fate in a faster and earlier fashion. And so from this data, they sort of conclude that the A and M together navigates a very different iCM trajectory and made us believe perhaps this ASCL1 is a naturally existing but more potent neuron version of HAND2. So isn't that nature is very fascinating? All right, coming back to the data, I have a few more key data to show you. Then I'll tell you the conclusion. So they also, well, here they use not only the single cell RNA-seq, but they also used a tag-seq. Basically, they did the dual omics. And they also determined a motif enriched by the A and M triggered reprogramming. And they find out around 740 mouse transcription factors change over the reprogramming trajectory mediated by A and M. As expected, the ASL1 and MAF2C, those motifs increased in their enrichment. And for the non-cocktail transcription factor, what they find is actually summarized here. So there is a gradual loss of the motif for AP1 complex in the accessible chromatin, while the motifs for a full spectrum of transcription factor families critical for cardiac development and function now became significantly enriched over the pseudo time. And you can see, you can tell the name, the BHLH, BZIP, and homeobox, and SMAD, and HMG, and TBOX. And if you look at the developmental biology literature, wow, those transcription factors are absolutely critical for heart development and function. But the MGD traditional cocktail were not able to really make these changes for the motif enrichment for all those transcription factors. And this is quite fascinating to us. And also worth noticing is that the motif of NKX 2.5 and is particularly gained. So here, so it's particularly gained in the A and M reprogramming because it never gained for the MGT reprogramming. Kind of offering this refreshing view about the historically missing cardiac transcription factor in the context of iCM reprogramming, because we never see like NKX 2.5, why this is always missing. And A and M actually really help to open the chromatin accessibility for the binding of this NKX 2.5. And TBX5 motif also gained in the A and M mediated reprogramming, in spite of the fact we remove it from the initial cocktail. So this is really quite powerful to open the chromatin structure, allow the binding of key transcription factors. And anyway, so this work really allow us to rethink about the direct reprogramming. And here we sort of kind of very brave to propose a new model for the future direct reprogramming, because we know the current reprogramming code is always using the lineage specific transcription factor and plus 1, 2, 3. You can see that the old cocktail for any kind of lineage always using the lineage specific one. But now with this work, we think maybe we can think slightly different. We can use this 1+1 model or the code, and instead of focusing on transcription factor normally expressed in the target cells during development, and we perhaps can use one potent binding factor, which could really perform a targeted opening of the chromatin, such as the ASCL1 in this case. That's why I mentioned in the middle of my talk, remember that our single cell dual omics find a very interesting phenomenon for the fibroblast, and it opened many BHLH binding motif. And they are very similar, keep that in mind. And within ASL1 overexpress, it can recognize in a less precise manner. So, and this model will just use a factor, allow for the targeted opening of chromatin, and where we can also supply another lineage restriction factor, and in our case is MAF2C, and that can allow the activation of target cell program. The 1+1 can further evolve to become a single hybrid or single chimeric protein model, where we can imagine we can pull out the binding motif from the potent binding factor and the important binding motif for the lineage restriction factor and make a chimeric protein. But it's easy to say, but not that easy to do, because we have to think about how we can make the regular folding and function always as how they perform in their original protein. But I think with all the new AI approach, especially AlphaFold, and with the prediction how those chimeric protein can fold, can save us time for the initial screening and just later on get the top candidate and move on to the experimental testing. So this is sort of the new data, new story, and we are very excited about, and hopefully can sort of change the way how people think about design cocktail or molecule for direct reprogramming in general. Coming back to the direct cardiac reprogramming, and there's still many areas, not only us, but other labs also continue working. And to summarize here, we believe we need to continue looking for our ideal or dream Tesla to identify the optimal fibroblast. And as I mentioned, we are very interested in this stage 2 cardiac fibroblast and generated all the in vivo mouse models and different knockout and pre-ER lines to further study that. And of course, the fibroblast heterogeneity become very different with different disease condition. We have to study that not only under MI, perhaps under other disease condition as well, and also during aging process and also with different genetic background. At the same time, we and others also continue refining this smart map and make this map more precise and hopefully can really tweak every single cell, well, not just the fibroblast in the heart. And ideally, this can be done also under any pathological condition and really make the personalized treatment into reality. Well, hopefully in the near future. Why I'm saying the near future? Because the field now is very, it's a great time and era to do science and to do the modern science, because we have all these newly developed approaches and technology available now, including the single cell multi-omics and spatial restoration with the AI machine learning, and also a very enjoyable high resolution imaging. And you guys also have a lot of expertise in this area with mathematical modeling. Of course, never forget about animal models, and especially those animal disease models, and with the facilitation of bioengineering and synthetic biology. I think the dream can be realized sooner than we expected. But last and not the least, I really have to give the credit to all the wonderful people I have the privilege and also pleasure to work with, like over the years, and many trainees here. And I hope I mentioned, if I'm not mentioned the specific name leading project, really apologize here, the previous member and also the current members in the lab, and also our wonderful collaborators and UNC core facilities and funding resources, AHA and also the NIHLBI. And thank you very much for your attention. And I really love to answer questions you might have. All right, I'll stop sharing here.
Great, thank you so much. Okay, so I don't see any questions in the Q&A yet, but I guess I'll get started with some questions that I have.
Sure. Yeah, I hope it's not too overwhelming. I try to provide the overview, more like give a history about how everything gets evolved. And I know a lot of people from different backgrounds, so don't worry about all the technical things. So I can explain
aspects of reprogramming clearly that are important. So it's good to hear about all of the different things that you're doing to try to address this problem. So my question was about the study where you looked at and you characterized the different types of fibroblasts, the different stages of fibroblasts, and I was wondering if that study was done in just the healthy heart, and then whether or not you think activation of myofibroblast plays a role in the reprogramming, or affects.
Yes. No, great question. So number one, so the published paper is just a healthy heart, and we start with the regular at basal condition, how the fibroblasts look like, and for a number of reasons, because also I know other labs are doing the single cell RNA-seq. We don't want to repeat the effort, and we always try to compare our data with, for example, Richard Harvey's data, because they publish in eLife for the MI heart what happened. And I know, like, also, I think meeting always wonderful. Always talk with like Jeff Moulton, like Jennifer Davis. Jennifer is very interested about how the fibroblast, myofibroblast transdifferentiation, and she is also using the single cell. I always, say to her, don't, we can always share the data. If she does the sequencing, we don't do it, but we can always share the data, try to compare what's going on. So currently for our lab, because we already did it at the basal condition, and we already pull out the candidate gene. So I would rather go the other route and focus on the candidate gene and look at them under disease condition and see how they change and how the three population dynamics change with the disease condition. And I also believe that will be a really nice complement to what Jennifer Davis is doing, maybe John Elroy is doing, like also Jeff Moulton. I don't know whether he is doing additional sequencing or not, but so then we have not sharing the data together, looking at the same question from different angles. But I think this is one of the very important area in the field now, and great questions.
Yeah, I think intuitively for me, it would be like a balance of trying to get regions. If you're talking about MI, want to get fibroblasts in the region of the scar, but that they're also responsible for scar deposition, but also need, like, it's regional, right?
Yes, exactly.
Which fibroblast populations are being converted. But okay.
Yeah. Also another problem. So the original actually remind me about the technical difficulty. So to go around about it, because when you think about the scar area, really to dissociate the cells and can induce a lot of stress to the cell, and then sometimes the signal will be overwhelmed by stress responsible signaling pathways. A lot of immune genes get upregulated. So we are thinking about how to solve the problem, but currently, well, theoretically, those single cell spatial transcriptomics will be helpful, but currently there's still a lot of limitation. I almost start to say that from a year ago, we are watching to see how the technology get improved, but even nowadays, the resolution is still not the single cell level. So I guess, wait until the point their resolution can be improved significantly, and the spatial restoration will be really, really helpful, and without dissociating the cell, because artificially will really bringing a lot of noise. But that's why the original, that question, the point, I think it's very important for us to consider also to address that with the single cell genomics approach.
Great. Okay, so yeah. I'll ask some questions from the chat before I ask.
Yeah, I see the hand from Robert.
Okay. Go ahead, Rob. You can go first. Oh, you're muted.
Thanks, Lee. Wonderful talk.
Thank you.
So I guess I'm going to ask a question from the translational perspective, and a couple of things that I was thinking about as you were talking. So the first thing is most of your work is focused in mouse. Yeah. So one wonders whether, how much of it is transferable to, say, a large animal like a pig that might be a stepping stone on the way to translating to humans, and then humans themselves. Yeah. And then the second part of my question is, what's the electrophysiological characteristics of these iCMs? Are they immature myocytes? Do they recapitulate mature myocytes? And the reason for asking that question, of course, is some of the stem cell work has shown that, yes, you can get embryonic stem cells to repopulate injured myocardium, but then you start to have problems with arrhythmias and so on. So I'm just wondering, what your thinking are on these practical translational issues.
Yeah, definitely. I guess we can talk more during our one-on-one meeting, but just I try to give more brief answer, because otherwise I will keep on talking for another hour. So I think number one, to answer your question, so when we do the in vivo cardiac reprogramming and we isolate those converted cells by following the linear tracer and using the FACS sorting to sort out the converted one with the lineage marker and demonstrate they're pretty similar to the ventricular cardiomyocyte, but keep in mind those are the in vivo reprogrammed cells. cell. And we believe the environmental cue play a significant role to sort of promote the maturation because they started to convert from the fibroblast at the very beginning, and later on more like a more natural process for them to mature in the live heart. So we demonstrated in that very early Nature paper already. So physiologically, all the patch clamp experiments, we also show even the calcium propagation from the non-reprogram 1 to reprogram 1, and they seems all pretty good, or actually decent. So I think much better than the cells. Actually, you differentiate them, like the stem cell, you differentiate them and then inject back to the heart. So we think our approach is more advantageous compared to the stem cell therapy, sort of transplantation approach. But to answer your arrhythmia question, that I have to say, so we need to be carefully monitor that because in our study, we only monitored heart for three months, not really longer than that. And there is pretty minimal arrhythmia because we think the cells were converted in situ, different from how the stem cell therapy, you introduce the cell and inject randomly, and it's very hard for the integration. So arrhythmia, of course, need to be addressed, like following longer term and also using large animal. So that's why I'm answering the question the back way. Large animal and human, right? So, all right, so the field currently is so divided into the industrial effort versus the academia effort. So it's quite interesting, but I love the field because we all communicate so well. So Deepa, Eric actually had a company called Tenaya Therapeutics because they are doing all the work in large animals, and they presented sometime at the scientific meeting as well. So to avoid repeated efforts, so we don't even propose to testing large animal, rather focus on the molecular mechanism and identify new facilitator and barrier, and hopefully the information can feed back to industry again. So I think maybe Deepa or Eric can give better description than me, but I think their large animal study are very, very promising, and they use the pig, and they use also like I think they use rat as well, but the pig for sure they use all the AAV approach to introduce their cocktails and find the functional improvement and scar size reduction in the pig model as well. How far away from the clinical implication? I think the two sides going in parallel. The company currently is still using transcription factor and probably the early one, but they are using the AAV, and that should be avoided in the future. But I guess they wouldn't stop trying to apply that in human patients, especially the late stage heart failure patient, right? So, but at the same time, hope the academia or the exercise can help them to improve as well. And in the future, it's not AAV virus, it's not transcription factor, can be replaced by more advanced version. I think the idea about the single one protein will be great, but I still think more need to be done. And the ASL1 plus MAP2C and in the future is chimeric, one single protein can be ideally delivered to the injured area. But that takes time, but I guess the company want to push using the less optimal, but still, pretty decent therapeutic approach and sort of converting the fibroblast into cardiomyocyte. But we can follow more on that. Later on.
Thanks.
Sure.
Okay, so some questions from the chat. Yassine Sassi asks, what do you think about direct reprogramming of cardiac fibroblasts to cardiomyocytes using non-coding RNAs?
Yeah.
Yeah.
So that's great point. That's why I think I love the field, because we're not competing. Okay. Victor Zhao's lab and his trainee, like his first author, like now also became a faculty. They are very dedicated for microRNA mediated reprogramming, and the full microRNA is 1, 499, 143, and what's the other 143? So anyway, they're all 208. So full microRNAs, they demonstrated can reprogram fibroblasts into cardiomyocyte. I guess the maturation is an issue for them, because we talk a lot and talking about how powerful the transcription factor, because once the transcription factor converted fate and maturation becomes less an issue. But for microRNA, it seems like they can turn on the program to some extent, and the cells are not sufficient to be mature by themselves. But in vivo situation will be different because the environments actually help. So a side topic is Victor Zhao actually also had a new company company started and proposing to use just the microRNA to reprogram the fibroblasts into cardiomyocytes. And so that's why, my lab per se, we are not working on microRNA, but we think with their efforts, probably one day it can merge, because for them, the challenge is really the late step, how to complete the reprogramming process, and the cells can be more close to the endogenous cardiomyocyte. But because obviously using the transcription factor, it happened more potently and more complete. So, but I think, with all the research they're doing, maybe getting close one day as well.
Okay. Another question from Libu Lee. Great talk. Can you comment on whether myofibroblasts I guess they can potentially transdifferentiate into immune cells, such as monocytes?
Oh, to immune cells. Not yet. So I think that's a great point. Number one, so first of all, at least based on what we heard from meetings and what other labs are doing, they're thinking about transdifferentiating myofibroblasts or differentiate fibroblasts into at least endothelial cell. And I know that area pretty active, and a couple labs are working on that. But so it seems like people haven't thought about changing changing fibroblasts or myofibroblasts into immune cell. And I guess because immune cell, the cell features and how it functions in the heart is very significantly different from the resident cells already in the heart. Because you can think about infiltrating immune cell and resident one, they play different role, and how the injury actually attract the more immune cell coming from the system. So I guess the simple answer is no. As far as I heard, and we are not working on that. We haven't seen people working on that yet, but people do think about myofibroblasts or fibroblasts converting into endothelial cells or parasites or other cell types. But not, not the parasite, not the epicell, the epicardium cell or endothelial cell. I noticed people are working on that, but not the immune cell yet. Yeah.
Okay, well, if there's no more questions, I guess we'll just thank you for your time, Lee, and I hope you look forward to the rest of your visit. It was great.
Yeah, sure. My pleasure. Yeah, thank you all for listening. Yes, I will see you guys later.
Yes.
I love the applause. Thank you.